This is Dr. Aria Vaishnavi from MD Anderson, and she is a researcher and assistant professor there. And I'm going to hand it over to her to get started.
Thank you. Thank you so much for the introduction, and thank you all for this opportunity to be here today. It's wonderful to meet you all. I'm very excited to be here and talk to you a little bit about my research. I just wanted to say I've never done something like this before, so I did my best to come up with a broad laydown of BRAF-mutant lung cancer, and I'll mention again, I'm not a physician, I am a researcher, so my lab studies BRAF-mutant lung cancer, but I've done my best to kind of compile some broad overview, also clinically, of what happens with BRAF-mutant lung cancer. And so if you have questions, just please write them down, let me know. And if I don't know the answer to your question, I'd be more than happy to help you find the answer by reaching out to one of my clinical colleagues or a friend or other researchers across the world to help you get an answer to your question. So with that, my name is Aria, and I just started my own research lab in October. So I'm a brand new assistant professor in the Department of Cancer Biology at MD Anderson. It's a great opportunity for me to engage with all of you. And I want to talk to you about BRAF-mutant lung cancer. Now, for just a little bit of background about lung cancer is a horrible disease. It's the global leader in cancer-related deaths. And now a fourth of all lung cancers are never smoker related. So it's a combination of smoker and never smoker disease. But there's also other ways that people get lung cancer, including radon gas, pollution, asbestos, chest radiation, genetic mutations, secondhand smoke, as well as coal burning stoves. 20 years ago, lung cancers were categorized and treated based only on the categories of histology, which you've probably heard your doctors talk about. It's how the cells look in your cancer, and they categorize it based on which cells are involved. But about 10 years ago, maybe 20 years ago now, we started to reclassify this disease as we identified two of the main molecular subsets of oncogenic drivers. Those are mutations in genes that drive lung cancer. Those two genes are KRAS and EGFR, which I'm sure you guys have heard about, not just in your group, but in general in the lung cancer spectrum. But a lot of progress in the last years have identified a heterogeneous breakdown of molecular events that drive lung adenocarcinomas, which is the type of histology that most often has BRAF mutations, but it can also occur in squamous cell carcinomas. And so this kind of shows us what the evolution of lung cancer subsets has evolved to over the last several decades. And so these are the known dominant oncogenes in lung adenocarcinoma. And I'm mostly going to talk to you today about BRAF-driven lung cancer. And that's about 5 to 10% of adenocarcinomas, depending on who did the breakdown of the sequencing relevance. But that percentage and that chunk of the pie is a huge patient population, as you guys are probably aware, because 1% of lung adenocarcinoma is about 2,500 people in the US every year. So 5%, that's a lot. That's over 10,000 patients. And if it's closer to 10%, it's even more. And the reason that this molecular classification is important is because a ton of these mutations or biomarkers serve as FDA-approved biomarkers to designated targeted therapies, which you've probably heard people talk about. A good example is dabrafenib plus trametinib, but also several emerging ones as well. And the reason this is important is because these patients respond better to these targeted therapies than they do to the standard of care, which traditionally has been platinum-based chemotherapy, but is kind of transitioning towards immunotherapy now. And this focus of targeted inhibition on dominant oncogenes is something that I've been working on in lung cancer for over a decade now. And I want to start by giving you a little bit of background about BRAF as a gene. And I'm going to try to keep this pretty broad, not get into too many nitty-gritty science details, but please feel free to let me know if this was difficult to understand. I'm happy to break it down a little bit more. So some things that you should know about the MAP kinase pathway, which is a signaling pathway that harbors BRAF and KRAS. It's a signaling pathway that's important in normal development as well as cell physiology. So we absolutely need it for embryogenesis and tissue homeostasis. Now, certain mutations in RAS or BRAF can promote developmental abnormalities. This is in addition to cancer, and we can call those RASopathies. But you can also get point mutations not in the germline, which activate RAS or BRAF. And those are implicated in the genesis and the maintenance of about a third of all human cancers. So it's a huge number of cancers that have mutations in these two genes, which are critical mediators of the MAP kinase pathway. So that's the take home from there. So mutations in BRAF are very frequent in human cancer. About 8% of human cancers have mutationally altered BRAF. And you can see a breakdown here. It's not just a lung cancer problem. This is a cancer problem. 66% of melanomas, papillary thyroid cancer, almost 50%, 20% of cholangiocarcinomas, 18% of colorectal cancers, and so on and so forth. BRAF, the gene, it's a serine threonine protein kinase, and it's involved in activating the MAP kinase pathway, as I just mentioned. And that pathway is important for cell cycle progression, cell proliferation, and blocking apoptosis. So when I say cell proliferation, I mean making cells grow. And so sometimes cells growing can be a good thing, but sometimes when your cells grow too much or uncontrollably, kind of like in a tumor, it's a bad thing. And so we need to figure out how to stop them from growing in an uncontrolled manner. So BRAF, this is a cartoon schematic of what BRAF, the protein, looks like. It's a 766 amino acid regulated signal transduction molecule. Now, in terms of the mutations that we see in cancer, the majority of them occur in the kinase domain. The most common one that we hear people talking about is V600E. And so that's the mutation that you're generally tested for in lung cancer. And a lot of the research models are on BRAF V600E because that's the most common BRAF mutation that occurs in cancer. However, only 25 to 45% of BRAF mutations in lung cancer are V600E. There are other BRAF mutations which are less well understood. Those are not treated with dabrafenib plus trametinib. So that's something important to think about, is that we don't really test for these mutations routinely as much as we do V600E, because you're not going to get a targeted therapy if you have one of those mutations. So this is the MAP kinase pathway that I was talking to you about. It's activated by several known growth factors and cytokines, and they act through receptor tyrosine kinase signals, and then also activating mutations in RAS, which you can see here, or BRAF, which you can see here in this signaling cascade. And the pathway kind of shoots a signal, a chain effect of phosphorylation from here to RAS, to RAF, to MEK, and to ERK. And that results in changes of cell fates, such as proliferation or survival. And now when BRAF gets mutated, it becomes constitutively activated. So it no longer depends on extracellular signals or factors, and it's not regulated anymore. It's always on. It signals uncontrollably. So that's what it's doing in V600E-driven lung cancer. BRAF is signaling constantly through this pathway, and it's sending too many signals to keep growing and for the cells to survive. Now, fortunately, our third condition here is what happens when we use targeted therapies against BRAF. So the current FDA-approved schematic is dabrafenib, which is a BRAF inhibitor. I'm going to talk more about that in a minute. And then also trametinib, which is a MEK inhibitor. So this strategy is called vertical inhibition, when you hit a pathway twice to really try to block it, because this is a very strong signaling pathway that's the most commonly dysregulated signaling pathway in cancer. So we try really hard using two different drugs to block the pathway and reduce cell proliferation and survival. There's a broad spectrum of BRAF mutations that occur in cancer. The V600 mutants that I just mentioned, including V600E, are called class 1 mutants. Dabrafenib plus trametinib and previously vemurafenib work against class 1 mutants. However, the majority of class 2 and class 3, I think there's 31 known other mutants that have been at least partially characterized that are not sensitive. And so it's really important in the future that researchers like me make an effort to understand these other mutations and get a better understanding of what happens with BRAF mutations in lung cancer, not just V600E. And so that's something that my lab is working on in the future. And so I want to talk a little bit about what happens in the clinic. And so I'm going to start with targeted therapies, and then I'm going to move on to immunotherapies. And so first, the targeted therapies in the clinic. And so first, this idea of oncogene addiction is the foundation for why we use targeted therapies and why they work so well. It's the foundation of the personalized medicine paradigm that so many of us focus our research on today. So 15 years ago, a really smart guy named Bernard Weinstein coined this idea that in spite of having multiple genetic alterations, which you can see here, A, B, C, D, cancers often become addicted to one particular alteration, which is A, our oncogene of interest. And that inactivation of that oncogene can cause the tumor cells to apoptose or differentiate, which is how we block them. So that's one gene to rule them all. In this case, we're talking about BRAF. And so there's a lot of examples of oncogenes that create this addiction where the cancer cells are addicted. One example are kinase fusions, which I have shown here in this cartoon schematic, which one of the most famous ones is BCR-ABL. You've probably heard people talk about that before, and if not, that's totally fine. But there's also BRAF fusions, which I heard come up in the pre-chat a little bit. But you can also just have activating point mutations like BRAF V600E. And at the end of the day, they constitutively activate a kinase, and that results in aberrant signals to proliferate and to survive through different signaling pathways like the MAP kinase pathway. And targeting these oncogenes with a kinase inhibitor can shut down these signaling pathways. So they were signaling, they were continuously going like the Energizer Bunny, and then our goal is for this kinase inhibitor to block that and reduce those signals of growth. And so this is the NCCN guidelines on what happens with lung cancer patients. And so non-small cell lung cancer is broken down into adenocarcinoma, or large cell, or squamous cell. So histology is still important. It still plays a role in determining what tests and what type of disease patients have. But the big thing here is biomarker testing is recommended for all appropriate patients with newly diagnosed stage 4 disease. And it can also be considered for those with squamous cell histology. And in addition to molecular testing, they're also testing through immunohistochemistry PD-L1, which is the biomarker that is used clinically for determining whether or not a patient will most likely respond to certain immunotherapies. So we've got our molecular biomarkers like EGFR, ALK, ROS1, and BRAF, but we also have PD-L1 staining to see how much PD-L1 is expressed in those cancer cells. The hope is that broad panel-based testing is used here because it's more efficient than testing for one biomarker at a time. They have a lot of these signaling next-gen sequencing testing panels, like Foundation Medicine has one, and there's a couple of other ones now, which is probably how a lot of you were diagnosed with the BRAF mutation. It's also important to note that there's kind of an urgency here between getting molecular testing and waiting to start your first line of therapy. It can be helpful if you've been diagnosed with a certain oncogene to be treated with that targeted therapy, but it's tough to wait when you're a patient. So it's totally understandable that you'd want a therapy to be given and treated with that while you're waiting for your molecular testing to come back. And then from there, depending on that testing results, patients are treated differently based on each of these molecular markers. This is important because the best therapeutic outcome is seen if therapy is targeted to one of these mutations, especially for EGFR, ALK, and ROS1. BRAF is not as responsive as EGFR, ALK, or ROS1 to its targeted therapy, but it's still better than the standard of care. And then this is just a chart from a review in a book chapter that's coming out soon that I'm writing with my old postdoc mentor, Martin McMahon. And I wasn't exactly sure where to put this in the talk, but I really like this table because it highlights a few different but really elegant points. It has all of the approved or advanced stage clinical testing biomarkers and their relative therapies. But it also shows you that our research paved the path. Lung cancer researchers paved the path for these therapies by studying them in mouse models or in our research labs. And so this is highlighting all of the mouse models and the research papers that resulted in the understanding and subsequent FDA approvals or late-stage clinical trials for each of these drugs. And there's a lot of them, not just for lung adenocarcinoma, but also for small cell lung cancer, as well as immunotherapy. And so using mouse models in research labs can be really beneficial for understanding these therapies. And that's why it's such a paradigm for personalized medicine. So for BRAF V600E patients, only the V600 codon mutations, these patients are supposed to be treated first with dabrafenib plus trametinib if it's identified. First, that's the preferred strategy. However, upon progression, they can switch to immunotherapy or a different combination of immunotherapy plus chemotherapy. Another note is that if this is not well tolerated, which is not really an area of expertise of mine because I'm not a clinician, but something I've heard a lot about, and that's why as a researcher, I'm constantly looking for alternate or better or well-tolerated approaches, because dabrafenib plus trametinib can be very difficult on the patient. And so if that patient is having trouble tolerating that combination, you can be treated single agent with vemurafenib or dabrafenib. So that's something to keep in mind when you're working with your doctor. And the next line of therapy after progression is kind of at your doctor's discretion, but you have a lot of options depending on whether you're an adenocarcinoma or a squamous cell of origin patient. There are 3 generations of BRAF targeted kinase inhibitors, which are used clinically. We've kind of evolved from the first one, which was vemurafenib. That was discovered using a scaffold-based drug discovery to identify inhibitors of the BRAF oncogene. It's a selective inhibitor of oncogenic BRAF. It's potent, it's highly selective of mutated BRAF V600E, not wild type V600E, wild type BRAF. And then inhibiting the kinase activity of BRAF prevents signaling through the MAP kinase pathway and additional cell proliferation. And now we've evolved from vemurafenib to dabrafenib and oncorafenib. Oncorafenib is used more commonly in melanoma, and dabrafenib plus trametinib is used more commonly in lung cancer. So dabrafenib is a competitive second generation BRAF inhibitor, which binds to its ATP binding pocket. And although it can actually inhibit wild type BRAF, it has a much higher affinity for the mutant forms of BRAF, which is really important for patients and not developing certain toxicities. And so this is kind of a famous photograph that some of you may have come across on the internet, but it's showing you that the number one problem with targeted therapies is acquired resistance. And so here we have a 38-year-old BRAF mutant melanoma patient who was treated upon identification of disease with vemurafenib. And you can see this incredible response. It's very visual. You can see that the patient responded incredibly. However, 3 months later, they became resistant, and you can start to see the disease recurrence. So while targeted therapies do work well for a while, a few months, anywhere from 3 to 10 months usually, eventually the patient becomes resistant, which is a huge problem and something that we're actively working on in the lab to improve our understanding of. And so my lab primarily works on targeted therapies against BRAF, but I also want to talk a little bit about immune checkpoint blockade in the clinic because it's important for lung cancer and it can be important for certain BRAF patients. And so I may not be the best person to answer your questions on this, but if you have any, please do let me know and I'd be happy to do my best to help you get an answer that makes more sense. So an important part of the immune system is its ability to keep itself from attacking normal cells in the body. So under normal circumstances, it uses checkpoint proteins on immune cells, which act like switches that need to be turned on or off to start an immune response. However, cancer cells sometimes use these checkpoints to avoid being attacked by the immune system. So we want to disable that from happening and allow the immune system to attack the cancer cells. And so we do that with a couple of different classes of immune checkpoint blockade. The first one is the PD-1 axis. So PD-L1 is a ligand for PD-1, which can signal to prevent T cell activation. So if we block it, we can activate T cells. Similarly, CTLA-4 is an immune checkpoint that downregulates immune responses. So if we block it, we can convince our T cells to attack the cancer. And so some examples are nivolumab and pembro, as well as atezolizumab and ipilimumab. Those are tough to say. Can't say that 5 times fast. But another important consideration with immune checkpoint therapy is whether or not a patient has high mutational burden. And this is just a picture from a review where they were talking about how different cancers tend to have different types of mutational burden in neoantigens. And that is a predictor of whether or not a patient might respond well to immunotherapy. So that's why it can be important. And you can see here that lung adenocarcinoma and lung squamous cell carcinoma are pretty high on the list. And it depends sometimes whether or not a patient has a smoking history or not. That can contribute to whether or not you're more responsive to immune therapy. So while first-line targeted therapies are the current standard of care for lung cancers that have actionable mutations, the cancer cells inevitably get acquired resistance. So immune checkpoint inhibitors have been able to improve the outcomes of metastatic lung cancer, but their efficacy long-term for patients who have targetable drivers is largely unknown. We're still in the process of trying to understand all of these things. So you can kind of see the breakdown of how we do things. We do the molecular testing for EGFR, BRAF, RET, etc. If a patient has no actionable mutations, they can go through IHC testing for PD-L1, which is that checkpoint inhibitor that I was just telling you about. And then based on how much PD-L1, they might get placed on a number of different immunotherapies, which are shown here in the boxes. And you can also have a combination of immunotherapy with chemotherapy, which you can see here, carboplatin or cisplatin with pembro or atezolizumab. So there's different combinations or options, which can depend on specific details of your disease, whether it's an adenocarcinoma or a squamous cell carcinoma, or your physician may have additional criteria that he uses to make that decision, he or she. But basically, these are the main options for patients with advanced or recurrent lung cancer. And it's a little complicated. This is something to consider for your physician to consider after you've progressed on dabrafenib plus trametinib, or if you're not responding to it. And this is just showing you the basket trials from the targeted inhibitors from BRAF. This is non-melanoma basket trials that were published a number of years ago to show that there's kind of a general trend of a mixed bag. Some patients do respond, some patients don't respond. So this is beyond just lung cancer is included, which you can see here. Overall, it's better than colorectal cancer or some of these others, but it's kind of a mixed bag. So while there was a lot of success with BRAF inhibitors in melanoma, it hasn't been as clean for some of the other BRAF mutant diseases. And this is just a recap. So generally speaking, patients with V600E BRAF mutations in lung cancer do respond to dabrafenib plus trametinib, and it's better than the standard of care, which was platinum-based chemotherapy, which you can see here. There are patients who had complete responses, which are shown in green, as well as partial. The majority of them had partial responses in this pink color. So this is a good option compared to the standard of care. So as a researcher, my lab is working on understanding BRAF mutant lung cancer. We want to use models in the lab that are relevant to human disease. So we have cell line and organoid 3D models that we make from mice or human tumors. We use mouse models that are genetically engineered to have the V600E mutation, and I'll talk to you a little bit about that model. We also use human-derived mouse models. They're called PDX models, or patient-derived xenografts, where we take human lung cancer patient tumors and we put them into mice and we study them. We try to understand what pathways are activated, how different therapies work, with the goal of ultimately improving patient care in the future. Traditionally, labs have focused on BRAF V600E, but I think it's really important to create new models to get a better understanding of what other BRAF mutations do as well. And then we can also use patient samples directly, not just to create models, but also to study different aspects of human disease. The better our models are, the more helpful they are at improving therapy and understanding what's going on in human lung cancer patients. So we use the BRAF cat mouse. It's based on the human V600E mutation. And so it's a Cre activated TD tomato, which is a fluorescent protein. And so that's why in the mouse, you see red fluorescent tumors, which allow us to trace them and track them over time. So normally this mouse has wild type BRAF, but we can give it virus, which expresses this CRE recombinase, which is a special enzyme, which allows us to initiate lung cancer in these mice. And it elicits these benign adenomas without an additional mutation. They start out as benign adenomas and can progress to fully malignant lung cancer. Then we treat the mice with the dabrafenib plus trametinib, which is the FDA-approved therapy of choice, which is what the human patients get. You can see where it hits the two genes in the pathway. And you can see that in the mouse lungs, those tumors are gone. So we can use this in our models in the lab, and it works similarly to how it works in a human. But like humans, if we stop using the drug or we continuously treat with the drug, eventually the cancer comes back. So we have good models in the lab which allow us to study this and mimic human lung cancer. And this is the type of experiment that we do in the lab. So we take the mice, we initiate BRAF V600E mutations, and then we treat them with either vehicle or dabrafenib plus trametinib. It initiates tumors. They get a little bit less tumors in the dabrafenib plus trametinib, but then we can sequence them and try our best to understand what changes happen when we use dabrafenib plus trametinib, what cells are left, and how we can prevent drug resistance from happening in the future. We can also use human PDX models, which come from BRAF-mutant lung cancer human patients. We take pieces of the tumor and we put them in mice to try to mimic human disease. And we can treat those mice as well, these human tumors, with dabrafenib plus trametinib or another candidate drug, which allows us to test other inhibitors in the research lab and compare them to dabrafenib plus trametinib. So our goal is to find other therapies that may work, that might be more well-tolerated or more potent than dabrafenib plus trametinib. So that's another area of investigation in the lab. And ultimately, our goal is to understand lung cancer at different stages. So we have normal lungs. We look when lung cancer first initiates. We look at what happens to maintain it. We look as it progresses to a more malignant form of adenocarcinoma. We also want to understand the treatment response, as well as lethal progression. And our goal in the lab is to use these models that mimic human disease as closely as possible with the goal of improving patient care. And so to summarize what I've told you, BRAF V600E is an actionable lung cancer oncogene, but we want to do better. We want to find better treatment options and use our models in the lab to understand this a lot better. And so with that, I'd like to thank the people in my new lab as well as my old lab. At the University of Utah and Martin McMahon's lab when I did my postdoc. His lab started the original BRAF studies in lung cancer, and now my lab is continuing that here at MD Anderson. And so with that, I'd love to answer any questions and talk to you guys a little bit more.
There are a few questions in the chat. So do they test for all BRAF mutations in the next generation test?
Most of them are targeted sequencing panels, which are looking for specific mutations. So generally speaking, you would not be tested for all BRAF mutations. You'd probably be tested for V600 codon mutations, but not some of the other common mutations.
Are all current BRAF targeted drugs the same? For example, dabrafenib, Braftovi.
So there's 3 different generations of inhibitors. They're slightly different. They're all BRAF inhibitors, but they're not the same. They're supposed to get better when we have a second and a third generation drug. It might be more potent. It might be more specific. It might be more well tolerated. There's usually a reason why it's a next generation inhibitor. But all 3 inhibitors are paired with a MEK inhibitor, whether it's trametinib or binimetinib or cobimetinib. There's usually a MEK inhibitor that's paired with it. So the combinations are similar, but not identical.
So you could potentially go from like dabrafenib to trametinib to Braftovi, Mektovi, and have a better response?
That is difficult to say. I don't know about better response, but you probably wouldn't go from being treated with one of them to another combination. If you progress on the BRAF-mutant plus trametinib, they're not going to put you on oncorafenib plus binimetinib. They're going to put you on immunotherapy. Because if you've progressed on BRAF targeted therapy, they're not going to give you a different targeted therapy that has the same targets.
Can BRAF fusions be detected through standard biomarker testing, or is RNA testing needed?
That's a great question. So RNA testing is the gold standard for gene fusion detection. So I worked on gene fusions for a long time and during my PhD. RNA is the gold standard for gene fusion detection. You're going to miss a lot of gene fusions if you use DNA. For BRAF fusions, they would have to be on a targeted panel. And so it depends on which panel you're using. A lot of the larger ones definitely do have BRAF fusions as a target, but I think we'd have to talk about a specific panel. And it's very possible to miss it with DNA-based sequencing and not RNA-based sequencing.
Can you explain, because I'm a little confused by RNA versus...
That's the type of nucleic acid that's isolated from your sample.
So it's still part of your biopsy.
Absolutely.
Ran differently.
It's a little harder to get RNA, high quality RNA versus DNA. DNA is a little bit easier to get. But I don't want to get too technical.
Right.
But RNA has a lot of advantages over DNA in detecting gene fusions. And the quality of the RNA is also really important. Whereas for DNA, you're more likely to have success with the sequencing than with RNA.
They found my fusion with RNA sequencing. That's why
I'm not surprised. Yeah,
that.
RNA is definitely better than DNA for gene fusions.
Is your lab doing research on BRAF fusions?
Currently, no. But that's definitely an area I'd be interested in going into or back into in the future. I've worked with ALK, ROS1, and NTRK primarily. That's what I worked on as a graduate student. And then a little bit during my postdoc, we worked on NTRK fusions too. And because BRAF fusions do occur, I would be very interested in making a mouse model or studying it in my lab in the future.
How common it is. I see a lot of EGFR, not a lot, but when I see a fusion, it's either, I think, ALK or EGFR that fuses over. Now,
ALK is most common. There's a lot of gene fusions in lung cancer, which is another thing that my lab is studying, why there's so many. But the most frequent gene fusion is ALK, followed by ROS1 and RET, followed also by NTRK. But there's also, there is a possibility of EGFR fusions in general, not so much in lung cancer, but there's also the possibility of MET fusions, EGFR duplications or other things. There's a lot of ways to make an oncogene, and lung cancer loves to do them all.
Have you seen any BRAF fusions? Because I haven't seen any BRAF mutations change, or people, I should say. I haven't seen anybody say I was BRAF and now I'm this.
It's a mechanism of resistance for a lot of oncogenic drivers. I have not heard the prevalence of BRAF fusions in lung cancer, so I don't know what percentage or how frequently it occurs, but it definitely occurs, and BRAF fusions occur in other cancers as well. So I think it's an understudied area of biology, and it would be really interesting to make some models and get a better understanding of BRAF fusions in general, because it's a potent kinase, which is what you need to make a gene fusion. So I think it would be very interesting to study that more.
I understand that there may be some patients who have KRAS, but may also have BRAF. I'm not sure if KRAS mutates to BRAF or vice versa, or something totally different. Can you explain what, if any, association there is between KRAS and BRAF?
Most of these oncogenic mutations are mutually exclusive. So that term means that you usually have one or the other, because there's thought not to be a survival advantage, because a lot of this is kind of getting into survival of the fittest. And that's kind of what these cancer cells are, right? So you have, it's less advantageous to have two different oncogenes in one cell. However, tumors are heterogeneous. So you might have one oncogenic driver in one area of a tumor and one oncogenic driver in another area of a tumor. So I've heard of that. One example is EGFR and KRAS. I've heard of tumors where they have EGFR mutant lung cancer in one area and then a KRAS mutant in another area. And so you can have multiple drivers, but it's less likely to have them together because they're activating the same pathway. They're both in the MAP kinase pathway. So the cancer's not getting as much out of both of them activated right next to each other. They would benefit more from activating a different pathway or getting a different driver. There's also the question of how much you're activating the pathway, which is something that people in research talk about a lot. It's like a Goldilocks zone. What's the just right amount of oncogenic activation to make a dangerous cancer cell? So that's something that people are definitely working on. But generally, I don't think that BRAF and KRAS occur in the same sample.
When I was diagnosed 4 years ago, BRAF was reported at only 1 to 2% of cases. Is the increase due to more cases or just being identified?
That's a great question. I think the incidence usually depends on who's responsible for telling you. You'll get that's why I usually give a wide range when I give it. I would say it's anywhere between 5 and 10. But I would definitely attribute that to detection. It's becoming more common now. It wasn't even on the panels up front before. So of course it wasn't detected routinely. And now that molecular testing is becoming standard and BRAF V600E is on more of those tests, yes, absolutely. That's why it's coming up more. But that's also why we don't see the class 2 and the class 3 mutations as frequently, because they're not being tested for as routinely as V600E is.
Are there any treatments in the pipeline that you are excited about for BRAF in clinical trial?
There's some studies that are happening, which are really preliminary, so I'm not going to say too much about them. But there's a lot of hope. There's a lot of people who are working on this, really smart people, and trying really hard to find alternate therapeutic options, which may work for patients, but it's all very rational based, right? We don't want to just pick something or throw something against the wall and hope it sticks. We're trying to understand the science behind it. And so that's why these mouse models are really helpful, because it allows us to really understand what's happening in that cancer cell, to understand what pathways initiate the cancer, what pathways are required for it to stay on, what pathways are required for it to progress. That gives us different targets based on different stages of diseases. And ultimately, using a combination of different models, like the mouse model, the human-derived PDX models, we can really get after which targets are more effective. And so there's definitely a lot of targets that people are looking at, but nothing that's advanced enough to talk about publicly yet.
For mechanisms of progression for the BRAF inhibitor, does it look like most patients have the same resistance mechanisms, or does it vary greatly?
It's a great question. There's a lot of variation. There's been some publications about mechanisms of resistance, specific mutations that occur, or splice variants, as well as some pathways that are just common mechanisms of resistance in lung cancer, like EGFR signaling, for example, is a common mechanism of resistance because there's a lot of EGFR, HER2, and other ERB family members in the lung. And there's a lot of the ligands are present. So it's natural to see that pathway get activated accidentally. And you also see other mechanisms are also pretty common. But it's less well studied than some of the other. So like ALK and EGFR mutant lung cancer, they have a lot more studies, like with kinase domain mutations and a more precise breakdown of resistance and the percentages that it occurs than BRAF does. There's not a lot of resistance models yet for BRAF mutant lung cancer.
What are some of the side effects that would cause one to need to stop targeted therapy?
That's a great question. I actually think a lot of the patients in this room could give you a better answer to that than I could. One of the reasons that people have toxicities is because the drug is inhibiting that target in normal cells as well as your cancer cells. So like, for example, I'm going to use trametinib as an example. If you block the MAP kinase pathway in normal cells, it can make you feel sick. So that's why a lot of times these targeted inhibitors can be very toxic. EGFR is another good example. Like if you target normal EGFR in the skin or in their stomach, it can give you rashes or diarrhea because EGFR signaling is really important in those tissues. So if you take any tissue that needs MAP kinase signaling and you block MEK, or you block BRAF and you inhibit that pathway, it's going to give you similar issues.
Is there any connection with radon and BRAF?
Yeah, so my lab also studies radon-induced lung cancer and is trying to understand mechanisms of radon-induced lung cancer. So far, it's a little early for us to know which drivers. That's one of the big questions we're looking at, is which drivers are linked to radon. We'll see. I'll keep you posted. We need a little bit more data before I can say that, but it's possible.